So we are moving now in my talk from dealing with a single species, with E. coli, in the talk by Gabriel, to looking at the whole pool of resistance determinants in a food or in a food processing environment, so what we call the resistome, the complete pool of resistance determinants in a given sample. Let me see if I can, yeah. So let me add the laser, okay. So in the last years or decades, it's becoming apparent that the AMR crisis should be dealt with this One Health approach, not only looking at the human community, that is what we, of course, care more about the resistant, the resistance crisis in our hospitals, and that also antimicrobials are used in other parts and the animal health and the environmental health should be also taken together with the human health. No, the microbials are also used in food production, in farms, also in crops, and this both the human community and the animal farms and the farming activities are influencing for sure the environment through the wastewater, through manure and other byproducts. Etc., with interlinks between the different compartments, and of course, food production also at the end will impact back on us. So the food chain is one of the main places where this One Health approach can be fully seen and fully studied. No? And of course, the use of antimicrobials, as I mentioned in farming, is the first factor that it comes to mind, when talking about why the food chain could be a reservoir of antimicrobial resistant bacteria. And this can be very easily seen when we compare production systems where the antimicrobial use is very different. This is an opportunity we had with some colleagues in the animal health department in our university to look at two production sectors in the swine industry, in the pig production industry, that are very different. We have the typical intensive white pig farming, that is almost everywhere in the world. And then we have in Spain this particular breed that is the Iberian pig, that is very specific organic extensive farming, that part of the year they are outside eating acorns mainly from these trees. And they have very different antimicrobial use. Here you can see in red, for instance, the antimicrobial use in total in intensive white pig farms, and for different families of antimicrobials. It's very small, but you can see the trends. And in green, you see the total antimicrobial use in these farms of Iberian pigs, very, very low antimicrobial use. And when we compare the resistome in this study, it was very apparent that that not only in the feces of the animals, but also in the slurry from the farms, also in the environments, from the pens, from the places where the animals are kept, we have a much more abundant resistome, much more different types of genes, especially those located in plasmids. When we talk about the white intensive pigs farms in comparison with the Iberian pig farms, in the slurry, in the environment, in the feces and in the slurry. And also the profiles, when we compare profiles of genes found in the different compartments, they are totally different between different types of samples, but also between different types of farms. So it's very clear that the use of antimicrobials is having an effect. And these antimicrobial resistant microbes that are entering probably through the use of antimicrobials in farms, in crops, etc., they are at the end arriving somehow, part of them, to the meals we eat. So this was a very nice story we could do in a meat cutting plant that allowed us to follow the colonization of the environments in the farm through the first 1 year and a year half of activity. So it was a cutting plant that was newly open to business, and we could sample environments the 2 or 3 weeks before animals were entering into the facility, and then for a few months in an intensive sampling, and then every 3 months in a more large scale sampling up to 1 and a year half, 1 year and a half time frame. And what we saw was very interesting. So if you see here in green is sampling points before the animals enter. So they were starting to proof the machines. They were doing some cleaning and disinfection of the facilities, but the animals were not still in the meat cutting plant. Then you see here in purple this short term, the first month or so, or month and a half of activity of the meat cutting plant already with a lot of carcasses going through the place. And then this is more the long-term effect in red. So what happens over the year and a half in future months, in more advanced months. So you can see a clear trend of increase in the total abundance of antimicrobial resistance determinants, not only in abundance, also in the richness, so how many different determinants in each sample were found. The profile also changed. And these were samples that were carried out very early in the morning every day that we went to sample. So this means the resident microbiome, so not really with the influence of the animals of that day of activity. But not only through culture-independent methodologies, we focus also some studies on the Enterococcus isolates. So we were isolating Enterococci, and what we found is that, for instance, if you look at resistance to at least 3 different antimicrobial classes, the percentage of isolates that were resistant before animals entered was around 5%, and as soon as the animals entered, this rises almost to 60%. So, of course, these animals are introducing microbes into the food chain, and this somehow can arrive to the meals. But how is that food associated resistance? So the foods we eat, how much diversity and different types of resistance markers they harbor. So we could do this in a European project called Masters that was coordinated by Professor Paul Cotter in Ireland, and where we were leading a task from León based on microbiome sequencing in food processing industries. And in this task, we were developing protocols for microbiome sequencing, and we were generating a lot of data of metagenome sequencing in foods and in the associated processing environments. We could do tailored samplings in 5 different countries: Spain, Italy, Ireland, Austria, and Iceland. We visited more than 100 industries from different production sectors. It's true that it's a bit biased towards cheese production, so a lot of our collaborators were mainly in contact with cheese producers, and we had less opportunity to have representatives from other types of industries, but we also had pure meats, fermented meat products, fresh meat, fish, minimally processed vegetables, and we were analyzing almost 2,000 samples from raw materials in products and food processing environments. This amount of sequencing data was analyzed with very different perspectives, and we were from León the ones responsible, together with Austria, responsible for analyzing the resistome of all these samples. So that was a very nice opportunity. And when we carry out resistome analysis in our lab, we normally do it at a multi-layer analysis. So when we do a metagenomic sequencing of a sample, truly is as if we were sequencing many genomes that are together in that sample. And of course, we obtain short reads. So we are doing the sequencing with Illumina. So we have these small 100 and something base pairs reads. And the first analysis that we do can be done with these reads. So we with these reads, we target different databases. In this case, the ResFinder database, could be others, of antimicrobial resistance genes, and we identify reads that harbor antimicrobial resistance determinants. This provides a quantitative information, because we can look at diversity of genes, we can look at abundance, of course it's a relative abundance, we can look at the families of resistances that we see, etc. But then we normally go into assembly, so we try to assemble these reads, making a puzzle now, and to arrive to higher chunks of DNA, of the several base pairs. And here, of course, some of these contigs, what we call contigs, will have antimicrobial resistance genes as well. And the good thing about this is that as they are longer, and in some cases they can be several several kbs, even hundreds of kbs, depending on how well the assembly went. So we could look into the background of the resistance genes. So we could try to infer taxonomy, mobility, are they plasmidic contigs or chromosomic contigs, etc. Of course, this has to be taken with some level of caution, because there can be false positives and false information coming from this. But we have included some controls, and we are conservative in our analysis for this. And then If we go deeper, we can put these contigs into bins and we can reconstruct genomes. These contigs are identified as coming from the same genome, and we can, with this, reconstruct a genome. And of course, some of these can also have antimicrobial resistance genes. When we go deeper in this line, we are getting more information on the background of the resistance gene, but we are losing a lot of resistance genes because some regions are difficult to reconstruct, are in short contigs that, for this reason of being conservative in our analysis, we don't consider here, or are not even included in the genomes. Here in the genomes, normally we only finish with the chromosomic resistance genes. So the deeper we go, we lose some information, but we get more details about the background of the genes. Okay, let's see what we saw in the different layers of analysis. So if we stop first in the read level, so the first striking thing is that more than 70% of all the known resistance genes, even if only in one sample, they are present in the food chain. In most cases, only one sample or a very few number of samples in a very low abundance, but as I say, almost 2/3 or more than 2/3 of all the known genes are found in the food chain. If we look at abundance, richness, we can see that in general, in meat production, we have higher abundance, higher richness of resistant genes than in dairy, and in dairy production in general a bit higher also than in fish and minimally processed vegetables. The issue here is, as I mentioned, that we had a quite biased data set, so a lot of dairy production, then meat, and very little vegetable and fish production. But this is the trend that we can see. Of course, we will need to go for more countries, more industries, because, for instance, in some of the sectors, like the fish, it was only coming from one of the countries. So this biased composition of the database can somehow affect the conclusions in this regard. Looking at the types of samples, we also see, and it's quite interesting as well, that the abundance and richness of resistance genes is higher in the processing environments, in the surfaces, food contact or non-food contact surfaces, rather than in raw materials and in end products. And end products are somehow in the middle. So it seems that the environment somehow can be serving as a source for some resistance determinants here, which is also quite interesting. Looking at beta diversity, so with this coordinates analysis, what we can see is that the profiles of resistance genes that we identified are influenced both by the production sectors. For instance, here in the meats, in the raw materials, for instance, the difference depending on meat production, dairy production, vegetables. So you can see that the resistance determinants are totally different. The profile of the sample is totally different depending on the sector. But also within each of the sectors, we see this effect of the type of sample. So the raw materials and products and processing environments are somehow also different, statistically different. In general, what we see is a dominance of genes of resistance to tetracyclines, beta-lactams, aminoglycosides. Tetracyclines very much into the meat sector. Beta-lactams, aminoglycosides are starting to be more relevant mainly dairy sector. We have some samples like the raw materials of the vegetable processing industries that are totally different to the rest, and including new determinants that are almost not found in other categories of samples. And we have some interesting observations like that some resistance determinants like the macrolides resistance determinants in dark green or the phosphomycins in orange are almost absent in raw materials, but then they are present in processing environments and are starting to be also quite relevant in the final product. So these are like examples of possible influences of the environment of the processing itself on the final product resistome. Within each, these are the most abundant resistant genes found. So if we look at categories, we have within each antimicrobial class, we have many different genes. Here, for instance, only 5 of the most abundant tetracycline genes. These differ also a bit between types of sample. For instance, in meat, TTL, whereas in dairy, TTM or TTS are more abundant. So, of course, these are specific for every gene has a different story, and we have a lot of information to dig into. But we see this kind of sector-specific and sample-specific changes. As I mentioned before, even if we have thousands of genes different genes identified in our samples, there were only a few 20 gene clusters that were considered as prevalent, so present in more than 10% of the samples with a good abundance. These are the ones that I present here. Also, you can see the differences between types of samples. Okay, let's go now to the contig information. So we go to the next layer of the analysis, and as I mentioned before, here we can look into the context. We had almost 10,000 contigs coming from more than 1,000 samples that had at least one resistance in those contigs. We kept only contigs large enough and with several coding sequences so that we don't do taxonomic assignation for very short contigs. Knowing as well that sometimes, and we saw that in our controls, that sometimes when you get a taxonomic assignation to one taxa, sometimes it's not 100% certain. But very often when they fail, it's to another species in the same genera. So let's say maybe you get an assignation to a Staphylococcus aureus, and maybe it's not a Staphylococcus aureus, and it could be a Staphylococcus saprophyticus or a Staphylococcus pequorum. And it was in a small number of instances that was shown to happen in our controls. But anyway, what we can see here from these assignments is that around 50% of these contigs carrying resistance genes could be classified at species level. And near 20% of those assigned were assigned to escape bacteria. So here we see in red the escape taxa, Acinetobacter baumannii, Staphylococcus aureus, Enterococcus faecium, E. coli, Klebsiella, and several Enterobacter. As you see, most of the assigned AMR carrying contigs were to the genus Staphylococcus or to the genus Acinetobacter. These were the main ones. But if we see that almost 20% were assigned to escape, that means that around 80% were not assigned to escape. So many other taxa, sometimes from the same genus, but others from many very different genus, like Brevibacterium or Rautella, were also identified as very relevant taxa. And here we only present the most abundant ones, but we have many other minoritary taxa that were contributing also to the resistome. Something interesting to look at is if we look at here by sector and type of sample, it's like in fish and vegetable processing, there were very little amount of taxa contributing to the resistome, and mainly Acinetobacter johnsoni and Acinetobacter lwofi. Whereas in meat and dairy processing, there were much more different types of taxa contributing, and Staphylococcus aureus started to be a very relevant one, and other Staphylococci were also contributing importantly. At contig level, we can also infer things about the potential for mobilization of these resistance genes. And what we predicted is that around 1/3 of all the resistance determinants we found were associated with mobile genetic elements, mainly because they were present in plasmidic contigs. So we see here, you can see here by antimicrobial class, this means aminoglycoside, beta-lactams, tetracyclines, etc. And here what we can see is that in some families like aminoglycosides or tetracyclines, majoritarily they were linked to mobile genetic elements, whereas beta-lactams, phosphomycins, and others, they were mainly in chromosomic contigs and not near any known mobile element, because we were looking also at integrons, transposons, insertion sequences, etc., etc. And you can see here the most abundant resistance genes and how they were linked or not to mobile elements in our dataset. When we look at the taxonomy of these contigs as a function of whether they were identified as mobile or not, we saw that in general, all those contigs that were assigned to escape taxa, they were predicted as being highly mobile, so plasmids or other insertion sequences. Here, of course, again, we have to take that into caution, because when you assign taxonomy to a plasmidic contig, it can be wrong. But sometimes the plasmids are linked to specific, they have a more narrow host range of hosts. In other cases, they have a more wide host of range of hosts. And when they have a wide range of hosts, probably the taxonomic assignation could be erroneous. Whereas when we look into taxa that are not escape, like the Staphylococcus aureus, Acinetobacter johnsoni, and others that are not here in this slide, they are mostly chromosomic resistant genes. So this is what we generally found. Okay, looking also into mobility, we thought that it could be also interesting to look into contexts that harbor together in the same context at least 2 resistance genes. So that means that these resistance genes are probably co-transfer, located together, probably in plasmids. And we saw that around 20% of all the contexts that carry resistance genes harbor at least 2 resistance genes. And I saw here the main patterns that we found, the genes that were together more frequently. We see here the number of times, whether they were in a plasmidic or in a chromosomic contig, in what type of processing rooms they were found, associated with taxa. For some genes, they are more often found co-occurring with other genes in context than isolated in context. And here are the ones that more often come together in context. They are mainly found, these co-occurrences, of course, in plasmidic context, but also, very interestingly, principally on processing surfaces and end products, more than in the raw materials. And generally, they are more often linked to escape bacteria. We could even found some examples of contexts like this, where we had like 7 different antimicrobial resistance genes, an integron predicted, even metal resistance genes, and this with a high homology with a plasmid previously found and described linked to E. coli. So there are interesting examples that we found. We also wanted to look in the genes more frequently found in context, considering their abundance, trying to see if we found some trends, some patterns in terms of similarity of abundance of genes depending on the sample type or the category of the processing room, etc. So we did this particularly for the meat sector and the dairy sector. That is where we have more data and more information. This is the example for the dairy sector. So here you can see by color the code for the type of rooms and type of samples, contact, non-contact, and the different processing rooms. And here you can see that we have like 3 different clusters. The first one are the cold rooms of the facilities that had a very particular pattern, very different from the other types of samples. Then we have here the raw materials, so the milk, together with the cheeses just after cheese making and before ripening, and the processing surfaces from the processing room, that is where the raw materials are transformed into the cheese. And then we have the other cluster, the third cluster, that includes the end product, together with the processing surfaces from the ripening, the packaging, so the latest stages of processing, the smoking in the cases they have, the drying, etc. So this is mainly due to the fact that some determinants that are very abundant in the raw materials, in the milk, they are still present in the cheese just after making, in the processing surfaces from the cheese making facility, but they are either absent or very low abundance in the final product and in the latest stages of processing. And the opposite happens with a number of genes that are at very low abundance in raw materials, so almost absent, and they dominate over the time in the processing line. We also wanted to know the possible sources that are impacting on the final resistome, and we follow a different methodology here that was to align contexts with a very high thresholds in terms of homology, so looking for large contexts that were almost identical in the end product here in the right, and in different type of samples from the same facility. So this refers to the same facility. And it was quite interesting and quite surprising to find that the raw material had very little influence in the resistome of the end products. So as you can see here in the cheeses, only some contigs carrying TTS in Streptococcus and some other minoritary taxa and minoritary genes. Whereas the packaging, the processing, and the ripening rooms, the environments here had a big influence on and contigs found in the product, in the end product after ripening were found also with high homology, very large homology in these rooms, and linked to this taxa that you can see here. Similar analysis we did for meat processing industries, and again we see some things that are quite similar here. The raw materials are totally separated from the other types of samples because they miss some of the determinants that then appear in the processing rooms and in the end products. And here we have also differentiation between the end products and the processing rooms that are close to the end products, like the ripening and the packaging rooms, etc. And on the other side, the rooms that are at the start of the processing here, and also the non-food contact surfaces of all the rooms. So it's also quite interesting. And again here, when we look at this alignment of contigs between raw materials and products and different sources, we see the same. So raw materials very little influence, only this gene linked to contigs assigned to Citrobacter, and the cold rooms, the packaging rooms, the ripening rooms, much more influence with these determinants and these genes. Finally, the third layer of analysis are the reconstruction of genomes. So of all the genomes that we could reconstruct, that were 10,000, only 700 contain at least one resistance gene. They were coming from 85 different genera, and they were very little resistance determinants. In some cases, they are very conserved. So, for instance, all the Rotella or Niticola genomes harbor almost all of them these three determinants. Whereas in other cases, only some of the genomes of the species harbor these determinants. But again, and this can be very, very well seen here, of all the genes that we found at grid level, we miss all this when we move to contig level, and then we miss even more when we go to MAC level. So the deeper you go into the analysis, you are losing determinants, and you are mostly finishing here with only the chromosomic resistance genes. Okay, so that said, what we kind of find and hypothesize here is that, of course, resistant microbes are entering into the food chain through the use of antimicrobials, probably in different steps, in farming, through the farmers, through the food handlers, in different ways, and can be colonizing. And then what happens is that many of the different selection pressures that happen in the industries, the use of biocides, the different pHs, water activities, temperatures during processing, during ripening, are somehow selecting microbes that are well adapted to these conditions and to these industries. These well adapted microbes probably are only a subset of the ones that are entering. So that means that sometimes the determinants we found in the processing and in the products are not totally linked to the raw materials. And that could be sometimes through co-selection. There are examples of possible co-selection between biocides resistant and antibiotic resistant. They could be just because of these selective pressures of these environmental conditions that are somehow leading to the multiplication and to the colonization only by some particular species that are able to grow a low temperatures to form better biofilms in the processing surfaces, etc. And this, of course, is somehow having a big impact in the resistome. Again, coming to my first slide and coming back to this One Health perspective, now in the lab we are in the process of looking at some of these environmental impacts and interconnections. We are involved in a project funded by EFSA, where the European Food Safety, where we are looking into the use of wastewater in different activities in the food chain for irrigation or for cleaning operations in the industries, and see what impact that have into the resistome. We are also looking into the utilization of byproducts from the farms, but also from the food processing industries for the fertilization of crops, etc. And this is a place where I think in the next few years there could be more information, and we should focus also into this interconnection between the food chain and the community through the environment as well. And with this, I finish. I just want to acknowledge all the collaborators, all the people in our group in the University of León, but all the collaborators in the master project that are, and in other projects that were currently involved, that are the main ones are highlighted here, and of course to the funding by the European Commission for master and by our ministry for other activities related to resistance and the resistome. And yeah, and that's it. Thank you.